Hey guys, today is our second lesson on the immune system, so the defense against infectious disease. Okay, so the first video I answered a number of people's question from the first lesson. So if you haven't watched that yet, make sure you watch it. I'm actually teaching it as if you have watched that already. Now we are watching. We're gonna so there will be three videos. The first one is to answer the first question. The second video that I'm doing now is essentially talking about blood clotting as well as arterial cirrhosis. And the third one, we're going to be talking about antibiotics and antibiotic resistance. So let's get started. Now, if you remember from our first lesson, the skin and mucous membrane that forms the barrier that is part of our non-specific immune system. The idea is to keep everything out. Okay. Now, because it is such an important part, we do have blood clotting mechanisms. So, any cuts on the skin, you'll have blood clotting. The idea is to make sure you patch up that barrier as quickly as possible. So, clotting fat. So, cuts in the skin have sealed by blood clotting factors. Their chemicals are released by platelets. Now, if you remember from last year, I think you guys learned about the blood system from last year. If not, you're going to learn it in this course. It will be SL material. So platelets in your cell are responsible for blood clotting. So these platelets will release clotting factor, which is a chemical signals, and then you have a cascade. When we say a cascade, it's a chain reaction. Now, one good thing about having a chain reaction is that one molecule passes a message to maybe three more molecules, and then three of these molecules pass their chemical signals to another three. So if you can see how this chain is going, 1 to 3 to 9, it's a very quick way to get a lot of cell involved to scale up the production, so to speak. So now this here, so injured tissue will release clotting factor, prothrombin activator, and calcium ions. This converts the blood protein activator converts a blood protein to thrombin. Now, thrombin will split fibrogen to form fibrin. Fibrin tissues form a mesh over the wound, trapping red blood cells and platelets. The bleeding stops. The clot hardens and becomes smaller. New cells grow to repair wound sites. Enzyme plasmin is released to dissolve the clot. Now, this might not mean much to you, so I found a video which kind of explained this quite well. Not this video. I will have to skip a little bit first, simply because I'm going to move myself over here. Some of the materials you don't need to know it as well. So 1.18. Okay. So I'm going to turn this up so hopefully you can catch that too. If not, it is on YouTube. That's the name for it.
At the site of vessel injury, the first platelets arrive to start sealing the wound. Simultaneously, the coagulation cascade with its various coagulation factors is activated. This involves two pathways, the extrinsic and the intrinsic pathway. Extrinsic activation begins with now exposed molecules of the vessel wall, such as tissue factor, which forms a complex with factor 7.
Now, I'm going to skip through a whole bunch of that because there's a lot of details which you don't need to know. This video is produced by Bayers, who makes aspirin and things. So you don't need to know a number of them. I'm going to fast forward to a certain part. 1.19. This is closer to where you need to hear. So,
okay. Further down the cascade, factor 10A in combination with 5A activate thrombin and induces the so-called thrombin burst. One molecule of factor 10A can catalyze the formation of 1,000 molecules of thrombin. These large amounts of thrombin cause the further activation of platelets and the enhanced formation of fibrin. Fibrin then forms strands, making up the mesh that stabilizes the platelet plug in an arterial clot and holds together the red blood cells in a venous clot. It can be concluded that the central role of factor 10...
Now we're going to pause here, but it is quite amazing to have all these animations now which illustrate all these things. So essentially, what we're talking about here, that forming a mesh, all that, that's pretty much what that video was talking about and showing. And as you can see over here, that's the mesh with all the stuff, red blood cells and platelets forming. Now, pause again, any question about this? That is a level of detail you need to know for SL. Now a link-up question to it. Since we're talking about blood clot, we should talk about arteriosclerosis and also blood clot in the coronary arteries. Now, normally when I teach this material, I also will repeat some of this material also again when we're doing the heart dissection. However, with things where they are right now, I don't know when we're going to do that heart dissection or whatnot. So we'll see. So, arteriosclerosis. So you're talking about building up of plaque. Again, you're talking about the artery itself. So you see normal artery look like this. When you have arterial cirrhosis, you are talking about a buildup of plaque around and material around it. So now again, thank you that there are so many good videos out there. So I found another one. This one. Now the language is a little complicated, but we're going to play it through and then we'll refer back to the notes, okay? So now this one is pathogenesis of arterial cirrhosis. Now pathogenesis, pathos mean in biology and in Latin means disease. Actually, maybe Latin means something else, but in biology, it means disease. Genesis means the process of generation. So how does the disease get generated when we're talking about pathogenesis, okay? So.
Welcome to another Patho video. The topic of this video is atherosclerosis. Atherosclerosis is a condition where lipid plaques form within the walls of arteries. It is a specific type of arteriosclerosis, which is the generalized condition of hardening of arterial walls, whereas atherosclerosis is specific to the buildup of lipids that cause arterial walls to narrow and restrict blood flow. Atherosclerosis can be especially dangerous for the heart, since it often affects coronary arteries, which supply oxygen and nutrients to the heart. In severe cases, when blood flow through the coronary arteries is cut off, a heart attack, also known as a myocardial infarction, can occur, where heart cells or cardiac myocytes are deprived of oxygen and may die. Normally, most of the low-density lipoproteins, or LDLs, pass through the endothelial cells by transcytosis from the blood and then enter body cells by receptor-mediated endocytosis to be used in normal cell processes. The progression of atherosclerosis begins...
I'm going to pause here for a moment. So lumen of artery, this is where the hole in the artery, where the blood flows through. This here is basically the wall of the artery, the muscle wall of the artery. So if we go back to that handout, you're talking about here, okay, this pink area.
It's when the endothelial cells of the arterial wall become damaged. This can be caused by hypertension, smoking,
Hypertension is also another word for high blood pressure. Okay? Hypotension is low blood pressure.
glycemia, and hypercholesterolemia, which is an increased number of LDLs in the blood. Endothelial cell damage increases the permeability of the arterial wall, allowing LDLs to enter the tunica intima. White blood cells, such as monocytes, normally move freely through the blood vessels and do not attach to endothelial cells as they stream past.
Now, monocytes, HL student, you'll learn it. Monocytes is a type of white blood cells, and I believe on the handout here we're talking about leukocytes. Yeah, phagocytes. Okay, so it's a type of white blood cell, leukocytes or phagocytes.
However, when endothelial cells are exposed to irritating stimuli or damage, they will express adhesion molecules that can capture nearby white blood cells. These white blood cells undergo morphological changes that allow them to flatten and squeeze between endothelial cells. This movement of white blood cells out of the bloodstream is called diapedesis. White blood cells are capable of producing free radicals. And when these free radicals come in contact with LDLs, oxidation occurs. Oxidized LDL particles are especially effective at attracting and activating white blood cells. White blood cells then engulf the modified LDL particles, which stimulates them to produce even more oxygen-free radicals. It becomes easy to imagine that an area of endothelial damage will lead to an accumulation of modified LDL particles and migrating white blood cells. A positive feedback situation begins to arise when accumulating immune cells and modified LDLs bring in even more immune cells and modified LDLs. Macrophages in the tunica intima start to engulf modified LDL particles. Ultimately, this leads to the production of a cell called a foam cell. A foam cell is saturated with LDL particles, and the excessive amount of lipid in the cell gives the cytoplasm a foamy appearance. Foam cells ultimately die and release their contents, which are then quickly engulfed by other nearby white blood cells. Please note that the animation shows smooth muscle cells, also engulfing LDL cholesterol. Eventually, the accumulating lipid from the processes just described and the fragments of dead cells produce an area with a lipid core that begins to form a plaque. Endothelial cells cover the plaque. The plaque accumulates calcium salts and more dead cells over time, and it will harden. This plaque in the arterial wall is atherosclerosis. If the endothelial cells over the plaque are compromised, blood clots can form on the vessel wall. Remember that healthy endothelial cells normally express inhibitors of clotting, but now since they are damaged, they no longer do this. Over time, ruptured areas of plaque may create a situation where an area of plaque may jut out into the vessel lumen. A clot that forms and attaches to the wall is called a thrombus. If the clot breaks loose from the arterial wall and floats downstream to even smaller vessels, it is called an embolus.
Yeah, that's usually really bad. There's something called embolism, which usually when these things get clogged in the smaller blood vessels, if it is in the brain, that could be fatal.
Here is an animated summary now of the pathogenesis of atherosclerosis.
Okay, before we watch the summary, let's look through the handout and see how much we need to know. So in this case here, we talk about fatty deposit of building plaque. We kind of see that. By the way, earlier, so everything is happening in this pink section. This darker red section, that's the smooth muscle wall. That's very much on the low bottom part, okay? So you're seeing pretty much everything is in the pink area here, okay? It is becoming narrower. We talk about the coronary blood vessels. If it cannot supply blood, that can lead to myocardiac infarction, a heart attack. Takes a long time. Multiple medium and large sized arteries. That's true. Can lead to a heart attack or large in the heart following the sequence of events. So the video talk about all these sequence of events already. They talk about the high blood pressure, damage to the arterial wall. They didn't talk about smoking and the other stuff. Knowledge for you, not necessary for tests. Phagocytes, white blood cells arrive, secrete growth factor, encourage growth of the fibrous tissue. The endothelium becomes more permeable to LDLs, start to build up. Combination of lipids and fiber deposits called atheroma cause occlusion, so blockage in the artery tissue supplied by this artery receive less and less blood. If the plaque damage, blood clotting is triggered. Talk about thrombus, the clot form at the site, coronary thrombosis. So if that happens in your coronary artery, it can potentially cause a heart attack, lack of oxygen, lack of ATP, uncoordinated contraction, fibrillation. Fibrillation is uncoordinated contraction. If you want to remember, like for example, in first aid or in those medical drama, when someone's heart stopped, you need to use the defibrillator and then say clear, and you said clear, and then zap that person. You use a defibrillator to get the heart to go back on a rhythm contraction. So uncoordinated contraction is fibrillation. Okay. So let's just watch that summary and as a review on that list, okay? Oops. Okay, so we see the LDL more permeable. We saw the white blood cells going in. That's a positive, positive feedback cycle, causing more of the plaque being formed and fibers being formed, the thrombosis, and then the embolism, which is the clot that is let loose. Okay, so any question about clotting and arterial thrombosis? So we're going to go into our next video, which talks about the antibiotics as well as antibiotic resistance bacteria. Okay.